So quickly, the outline of the talk, the motivation of the work. I will present quickly the device and technology that we have used for this work, and talk through the experimental results that we got in a fluid medium and also in air, and briefly go through the future work on this research. So the motivation is to develop an acoustic sorting device that would give, that is tunable and that also allow better spatial resolution for particle sorting and also get high priority in the separation of the subpopulation. So the idea is to develop a dynamic acoustic field that allow us to take the advantages of the primary acoustic radiation force and the viscosity or the drag force. So when we look at these two forces, then we can see that from the acoustic radiation force, then this force is dependent of the volume of your particles and also the density of the particles. So our acoustic dynamic field takes the advantages of the size dependency of the microentities being sorted, as well as their density and compressibility. So the idea is to develop a dynamic acoustic field where we can ramp the phase of one of the transducer, and the particle will move with the Thank you. So this is the dynamic acoustic field. So the idea is to ramp the phase of one of the transducer and have a resting time where the particles can rather move to the next node or go back to their initial node. So for example, if we have large particles and small particles in a medium, heterogeneous medium, then the large particles will move with the field dynamic, while the smaller ones will just stay at their initial position. So it's just a cycle of this ramping phase and resting phase. Oops, sorry. Okay. So now let's go through the results in liquid. So the device used for that is a device that we already used previously for acoustic tweezing. It's a multi-transducer Petri dish device. And for this work, we only use two transducers here. So the transducers are PZT plates with a matching layer, alumina matching layer, plugged into flex circuits. And the flex circuit is folded into an octagon device. So how it works is the transducer works on two traveling waves, creating a standing waves, and we can shift the phase of one of the transducer, and the particles will move with the phase shift. Oops. Okay. We have also, in terms of electronic, high-speed amplifier, and we control the device with a LabVIEW interface where we can add our ramping time, but also the steps of the phase shift and the resting time. So to have a little bit an understanding or to have a hint about how the particles are going to behave into this dynamic acoustic field, then we developed a simulation program. And this simulation program is based on Verlet algorithm. And this Verlet algorithm allows us to put different size of particles and also streaming, non-streaming, changing the viscosity of the medium, etc. And when we enter our parameter, then we just put our dynamic acoustic field, then what we see is that the large particles shift from node to node while our small particles stay at their initial position. So really how it works is we push the large particles or the particles that we want to extract from the heterogeneous medium, and we just push them enough to pass the antinode, and that is the ramping time, and then we just leave it to rest, the resting time, where the large particles move to the next node while the smaller particles go back to their initial position. So we just need to push them enough to go to the antinode, and then they will just move to the next node during the resting time. And this is what our results, this is done via IMG. So we just put, it's an overlay of picture in IMG. And what we can see is our large particles here is 45 and 10 microns. So the large particles of 45 microns move with the field while the smaller particles stay in place. And this is a movie of the experimental results. So you can definitely see that the large particles of 45 microns move with the field while the smaller one stay in their initial position. What we also did is that we created software to be able to analyze our particles movement. And the software first identify where the particles are at the nodes, and then analyze the average particle displacement. And again, what you can see here is that your large particles in red move with the field, and this is like T-ramp, so they just move and then rest at the next node, and then move again to the next node and rest. And while the smaller particles that are in black just try to move with the field and go back to their initial position. So, and then we looked at the separation performance with, of course, calculating the efficiency of the sorting and the purity of the sorting. So again, we compared our simulation program with our experimental data, and what we can see is that there is a good match between the two data, and of course, the simulation program is binary, is good or bad, but we need to do the experiments to be able to have a better understanding or better analysis of the sorting, depending the different parameter of the ramping time and the resting time. For 10 and 45, we were capable of achieving 100% purity in our experimental data. Then what we were interested in is to look at smaller gap of particles in terms of sorting. So we tried to sort 6 and 10 microns. And once again, we were capable of achieving quite a good results with an efficiency of 97% and a purity of 97%. And once again, we reduce the gap between 8 and 10 microns in the heterogeneous medium. And again, we were capable of sorting the 10 microns from the 8 microns. Oops. Okay. We were also interested at looking at density separation. And for that, we put 10 microns particles with iron oxide and that are more denser than polystyrene particles. And what we can see is that the denser that are iron oxide particles move with the field, while the less dense particles, polystyrene, stay in their initial position. So in red is the iron field particles, and in black is your polystyrene particles that stay at their initial position. And again, we were capable of achieving very good results in terms of particle density separation with separation performance of 99% with a purity of 100%. So with this method, we are capable of sorting different size of particles, but also dense or less dense particles. The other thing that we were interested with this method is to look, to see if we could apply it to life science application. And we chose to look at separating dorsal root ganglion that is for regenerative medicine. So the main problem with extracting neuron cells from the heterogeneous medium is that when you use a fecal gradient protocol, you have a very poor recovery of neuron cells. So we were interested if by using our dynamic acoustic field, we could achieve a better recovery. So what you can see is that when we put the heterogeneous cell soup, if I can call it like that, then all the cells from the neuron, the DRG, the neuron cells and all the debris align within the field. And when we start shifting the field, then what you can see is that the neurons will move with the field, while the rest of the cells, the debris, the myelin cells will stay in their initial position. So once again,
it's for Q&A. Time is for Q&A now.
Okay, you could have said that 1 minute before, then I would have gone a little bit faster.
Okay.
So, well, I don't have time to show that. Sorry.